In this video, we will look at the structure of nephron and the process of urine formation. Nephron is the functional unit of kidney and it consists of glomerulus, Bowman capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule and the collecting duct. Blood vessels that are associated with nephron are glomerulus, which are located at the Bowman capsule, peritubular capillaries that surround the proximal and distal convoluted tubule, and vasa recta that surround the loop of Henle. Next, we will look at the process of urine formation. There are three main processes in urine formation, which are ultrafiltration, reabsorption, and secretion. Ultrafiltration is the first step in urine formation. It involves the glomerulus and the Bowman capsule. In ultrafiltration, hydrostatic pressure inside the glomerulus forces components from the blood plasma to move out of glomerulus into the Bowman capsule to become the glomerular filtrate. Components of blood plasma that will move out include water, vitamins, minerals, and many more. Large components such as blood cells, platelets, and plasma protein will remain inside the glomerulus because of its large size and the structure of Bowman capsule. Glomerulus is encapsulated by the Bowman capsule. The inner cell of the Bowman capsule are called the podocyte, and podocytes have cellular extension known as pedicels. The matrix between the podocyte and the glomerulus membrane are called the basement membrane. This structure ensures that blood cells, especially the white blood cell, will not be forced out of the glomerulus. Factors that increase efficiency of ultrafiltration are high hydrostatic pressure. These pressures are due to the difference in diameter of the afferent arteriole and efferent arteriole. High hydrostatic pressure formed in the glomerulus will force components out of the blood plasma into the Bowman capsule. Next, the highly coiled and long structure of the glomerulus will increase surface area for ultrafiltration. This results in high quantity of glomerular filtrate formed. The high permeability of glomerulus membrane, which is only one cell thick, help in component to easily move past the membrane into the Bowman capsule. So these three factors will help increase the efficiency for formation of glomerular filtrate. Next is reabsorption. Reabsorption is a process in which substance in the filtrate is reabsorbed into blood capillary. This occurs at the proximal convoluted tubule, descending and ascending limb of loop of Henle, distal convoluted tubule, and the collecting duct. At the proximal convoluted tubule, 100% of the glucose, amino acids, and vitamins are reabsorbed into peritubular capillary by active transport. Most sodium chloride ion are also reabsorbed. Bicarbonate ion and potassium ion will move into the capillary by passive transport. Due to the movement of this substance into the peritubular capillary, water potential inside the capillary will decrease. So, water will move by osmosis from the filtrate into the capillary. Next, the descending limb of loop of Henle are only permeable to water and impermeable to ions. So, only water will be reabsorbed here. While the ascending limb of loop of Henle are only permeable to sodium chloride ion and impermeable to water. Sodium chloride will move by passive transport at the thin segment of the ascending limb and by active transport at the thick segment of the ascending limb into the vasa recta. At the distal convoluted tubule, sodium chloride and bicarbonate ion will move by active transport into the capillary. Water will move by osmosis. And at the collecting duct, sodium chloride are reabsorbed by active transport and water by osmosis. Next, we will look at secretion. Secretion is a process in which substance from blood capillary is secreted into filtrate. This occurs at the proximal convoluted tubule and distal convoluted tubule. Both secretion and reabsorption process happen at the same time. At proximal convoluted tubule, hydrogen ion, drugs, and toxin are secreted by active transport into the filtrate. Ammonia and water will be secreted by passive transport. At the distal convoluted tubule, active transport of hydrogen ion and potassium ion from the peritubular capillary into the filtrate occur. Factors that will increase efficiency of both reabsorption and secretion process are: first, the tubule wall is only one cell thick. This makes the movement of substance into and out of the tube easier. Next, the inner surface of the tubule that faces the lumen has microvilli. This increases the surface area for reabsorption. And the epithelial cell that form the wall of the tube contain mitochondria. This is to form energy for active transport of substance. So, in urine formation, the first step is ultrafiltration to form filtrate. Filtrate will move to proximal convoluted tubule, where most of the reabsorption and secretion occur. Then flow into the descending limb of loop of Henle for reabsorption of water and reabsorption of sodium chloride at the ascending limb. No secretion occur at the loop of Henle. At the distal convoluted tubule, some reabsorption and secretion occur, and lastly into the collecting duct for reabsorption of water, salt, and urea. The filtrate that is left over from the reabsorption and secretion process will move into the bladder, where it becomes urine.